So good evening everyone. Today we are going to revise DNA replication. So what DNA replication exactly is? So replication का मतलब ही क्या है कि DNA की आइडेंटिकल कॉपीज़ बनना, ठीक है. सो इट इज़ जनरली फॉर्मेशन ऑफ आइडेंटिकल कॉपीज़ ऑफ DNA. अब DNA रेप्लिकेशन में 3 मॉडल दिए गए थे, सेमी कंजर्वेटिव, कंजर्वेटिव मोड एंड डिस्पर्सिव मोड. बट यहाँ पर जो मोस्ट एक्सेप्टेबल मॉडल है दैट इज़ योर सेमी कंजर्वेटिव मोड ऑफ DNA रेप्लिकेशन. उसमें क्या बोला गया कि हर एक पेरेंट स्ट्रैंड अपने कॉम्प्लीमेंट्री एक न्यू डॉटर स्टैंड की सिंथेसिस करेगा. सो दिस इज़ ऑल अबाउट सेमी कंजर्वेटिव मोड ऑफ DNA रेप्लिकेशन. नाउ DNA रेप्लिकेशन इज जनरली बाई डायरेक्शनल इन नेचर. एक्सेप्शनल है यहाँ पर कि DNA रेप्लिकेशन यूनिडायरेक्शनल किस केस में देखने को मिलती है, दैट इज़ इन द केस ऑफ माइटोकॉन्ड्रिया या वायरल DNA रेप्लिकेशन. बेसिक स्टेप्स जो हैं यहाँ पर वो है इनिशिएशन, इलॉन्गेशन एंड टर्मिनेशन. होता क्या है, इन जनरल एप्लीकेशन स्टार्ट्स होती है आपकी एक साइट पे. उस साइट को आप बोलते हो ओवरआई साइट वेयर एप्लीकेशन टेक्स प्लेस. हेलिकेज आपका डीएनए को अनवाइंड करता है. सिंगल स्टैंड स्टेबलाइजिंग प्रोटीन आते हैं वो उस अनवाइंड डीएनए को स्टेबलाइज करके रखते हैं ताकि वो फिर से रीफोल्ड ना हो सके. फिर प्राइमर सिंथेसिस होती है. शॉर्ट आरएनए प्राइमर्स बनते हैं ऑन द बोथ स्ट्रैंड ऑफ डीएनए. ये प्रोड्यूस करते हैं फ्री 3' OH. डीएनए पॉलीमरेज सिंथेसाइज करता है न्यू डीएनए इन द 5' टू 3' डायरेक्शन. ये बनाता है लीडिंग स्ट्रैंड जो कि आपके कॉन्टिन्यूअस स्ट्रैंड है एंड देन लैगिंग स्ट्रैंड दैट आर डिसकॉन्टिन्यूअस स्ट्रैंड्स. उसके बाद होता है प्राइमर रिमूवल. सो गैप फिलिंग एक्टिविटीज़ डन बाय डीएनए पॉलीमरेज. लाइगेज एक्टिविटीज़ डन बाय ligase enzyme. What's the function of ligase here? It seals the sugar phosphate backbone of the DNA. Then the last step we have termination step. We have replication fox meat. Now next है हमारे पास enzymes. So different enzymes जो कि function करते हैं in the prokaryotic and eukaryotic DNA replication में. So first enzyme है helicase. So prokaryote के केस में we have DNAB. In the case of eukaryote helicase का function करता है MCM 227. Helicase loader. So when we will talk about helicase loaders, in the case of prokaryotes we have helicase loader DNAC, but in the case of eukaryote we have no different helicase loader, so it remain as it is same. Third is topoisomerase. So in the case of prokaryote we have topoisomerase 2, that is DNA gyrase, and in the case of eukaryotes we have topoisomerase and 2 both. Now next is single strand stabilizing protein. So it is as it is in the case of prokaryotes. In the case of eukaryotes it is mainly replication factor A. Next we have primase. So in the case of prokaryotes we have primase DNA G, and in the case of eukaryotes we have primase DNA polymerase alpha. Now DNA polymerase. So the major replicative DNA polymerase in the case of prokaryote is DNA poly 3, and in the case of eukaryote we have epsilon and delta. Epsilon here leading strand synthesis, and delta what will do? Delta form the lagging strand, that is discontinuous strand. Lagging strand is known as discontinuous strand, that is the formation of Okazaki fragment takes place. Next we have primary removal activity. So in the case of prokaryote this activity is done by DNA pol 1, and in the case of eukaryote this activity is done by RNAs H and FEN, that is flap endonuclease. So major is generally FEN used here in the case of eukaryotes for primary removal activity. Next we have ligase. So yes, ligase in the case of prokaryote is simple as ligase, and in the case of eukaryote it is ligase 1 mainly. Clamp protein in the case of prokaryote is beta, that is used for DNA processing, and in the case of eukaryote we have PCN ring. Clamp loader in the case of prokaryote is gamma, and in the case of eukaryote we have replication factor C. Now if we talk about fidelity, that is accuracy with which DNA replicates. So अगर हम without proof reading लेके चलते हैं तो आपको एक एरर मिलता है पर 10⁵ न्यूक्लियोटाइड्स पे. बट व्हेन प्रूफ रीडिंग इज डन देन वी हैव वन एरर per 10⁻9 or per 10 to the raised to the power 10 nucleotide. Now if we will talk about DNA polymerase types, so in the case of eukaryote we have two type of DNA polymerase epsilon and delta, that will add 100 to 200 nucleotide per second. These polymerase will do 5' to 3' polymerization activity and 3' to 5' exonuclease activity. Here 3' to 5' exonuclease activity is known as proofreading activity. In the case of prokaryote we have 5 type of DNA polymerase, that is DNA polymerase 1, DNA polymerase 2, DNA polymerase 3, DNA polymerase 4 and DNA polymerase 5. Okay, so DNA polymerase 1 is unique and abundant because it has all three activities, that is 5' to 3' polymerization activity, 3' to 5' exonuclease activity, that is known as proofreading activity, as well as 5' to 3' exonuclease activity, that is primer removal activity. Now what will do DNA polymerase 2? base accretion repair. DNA polymerase 3 is a major replicative enzyme and at 1000 to 2000 nucleotide per second. DNA polymerase 4 and 5 will do SOS, that is TLS repair mechanism by damage, bypass damage or translation repair synthesis. Now if we will talk about inhibitors, in the case of eukaryotes we have different inhibitors. Some of the main inhibitors like irinotecan, atiposide, doxorubin. In the case of prokaryotes we have inhibitors like norflexin, ciprofloxacin, quinine. If these inhibitors are there, so there is no replication initiate. So what will do these inhibitors in the case of prokaryotes? They will do intra chain break. So this is all about the replication.